Welcome friends to a new session on genomics and proteomics. In this session, we will discuss microarrays. Introduction to microarrays. Molecular biology research evolves through the development of the new technologies used for carrying them out. It is not possible and sometimes impossible to research on a large number of genes using traditional methods. A microarray is a multiplex lab on a chip. It is a two-dimensional array on a solid substrate, example a silicon thin film cell or glass slide that assays large amounts of biological material using high throughput screening, miniaturized, multiplexed and parallel processing and detection methods. The concept and methodology of microarrays was first introduced by Sei and Chang in 1983 in Antibody Microarrays or Antibody Matrix in a scientific publication. There are different kinds of microarrays: DNA microarrays or microarray for DNA, which includes cDNA microarrays, oligonucleotide microarrays, BAC microarrays and SNP microarrays. chips for surveillance of microRNA populations. Protein microarrays or protein chips, which are microarrays for proteins used to track the interactions and activities to determine their function. Peptide microarrays. A peptide microarray or peptide epitope microarray or peptide chip is a collection of peptides that are used for detailed analysis or optimization of protein-protein interactions. Tissue microarrays. Tissue microarrays or TMAs consist of paraffin blocks in which up to 1000 separate tissue cores are assembled in array fashion to allow multiplex histological analysis. Cellular microarrays. A cellular microarray, cell microarray or transfection microarray is the multiplex interrogation of living cells on a chip spotted with varying materials such as proteins, lipids or antibodies which can interact with the cells, leading to their capture on specific spots. Chemical compound microarrays. A chemical compound microarray is a collection of organic chemical compounds that are spotted on a glass or plastic surface. Antibody microarrays. Antibody array, microarray for antibodies to detect antigens. Carbohydrate arrays or glycoarray, that are microarrays for carbohydrates. Phenotype microarrays, microarray for phenotyping of cells. Reverse phase protein microarrays, microarrays of lysates or serum. Interferometric reflectance imaging sensor. Interferometric reflectance imaging sensor or IRIS or the spectral reflectance imaging biosensor SRIB is used as a biosensing platform capable of high throughput multiplexing of protein-protein, protein-DNA or DNA-DNA interactions without the use of any fluorescent labels. The sensing surface is prepared by robotic spotting of biological probes that are immobilized on functionalized Si or Si oxide substrates. DNA microarray. DNA microarrays or nucleic acid arrays are a group of technologies in which specific DNA sequences are either deposited or synthesized in a 2D or sometimes 3-dimensional array on a surface in such a way that the DNA is covalently or non-covalently attached to the surface. So, we can analyze the expression of many genes in a single reaction quickly and in an efficient manner. DNA microarray technology has empowered the scientific community to understand the fundamental aspects behind the growth and development of life, as well as to explore the genetic causes of anomalies occurring in the functioning of human body. A typical microarray experiment involves the hybridization of an mRNA molecule to the DNA template from which it is originated. Many DNA samples are used to construct an array. The amount of mRNA bound to each site on the array indicates the expression level of the various genes. This number may run in thousands. All the data is collected and a profile is generated for gene expression in the cell. A microarray introduced in 1995 is a chip printed with thousands of variants of a particular polymer such as the cDNA, DNA or RNA. Each variant appears in a tiny dot, probe or spot containing many copies of the same sequence. The target sequences labeled with fluorescent tags are washed across the chip and the dots, when bound by the target sequences, fluoresce as shown in figure 1. The luminescent intensity of each probe indicates the relative quantity of the target present. A recent important application of microarrays is the tiling array, where nearly every sequence in a genome of at least some particular length, example 30 base pairs, is tiled on the array. Tiling arrays are powerful for genome-wide investigations and have many applications such as transcriptome mapping, protein binding sites mapping, etc. Various applications of microarrays end up with the same output format, that is, a high resolution image of thousands of variously illuminated little dots. A DNA array is used to probe a solution of a mixture of labeled nucleic acids and the binding of these targets to the probes on the array by hybridization, which is then used to measure the relative concentrations of nucleic acid species in solution. An array can be used to quantify an arbitrary large number of different nucleic acid sequences in solution. There are different methods of quantifying different nucleic acid sequences in a sample, including quantitative PCR, digital PCR and hybridization to optically tagged probe sequences. History of DNA arrays. Grunston and Hogness created the DNA array with the colony hybridization method. They randomly cloned the DNA of interest into E. coli plasmids that were plated onto agar Petri plates covered with nitrocellulose filters. Replica plating was used to produce additional agar plates. The colonies on the filters were then lysed and their DNAs were denatured and fixed to the filter to produce a random and unordered collection of DNA spots that represented the cloned fragments. Hybridization of a radio-labeled probe of a DNA or RNA of interest was used to rapidly screen thousands of colonies to identify clones containing DNA that was complementary to the probe. This approach was adapted to create ordered arrays by Jergen in 1979, who picked colonies into 144 well microplates. They created a mechanical 144 pin device and a jig that allowed them to replicate multiple microtiter plates on agar and produce arrays of 1728 different colonies in a 26 by 38 centimeter region. An additional transfer of colonies to the squares of Whatman filter paper, followed by a growth, lysis, denaturation and fixing of the DNA to the Whatman filter paper, allowed the production of DNA. During the next decade, filter based arrays and protocols similar to these were used in a variety of applications, including identifying SNPs, cloning genes of specific interest, cloning genes that are differentially expressed between the two samples, and physical mapping. Hans Leyrac's groups automated these processes in the late 1980s and early 1990s by using robotic systems to rapidly array clones from microtiter plates onto filters. The concomitant development of cDNA cloning in these periods, combined with international programs to fully sequence both the human transcriptome and human genome, led to create reference sets of cDNAs and cDNA filter arrays for human and other genomes. By the late 1990s and 2000s, sets of non-redundant cDNAs became widely available, and the complete genome sequences of some organisms allowed for sets of PCR products representing all the non-open reading frames in small genomes. These sets, combined with readily available robotics, allowed individual labs to make their own cDNA or ORF arrays that containing gene content represented the vast majority of genes in a genome. The birth of the modern DNA array. In the late 90s and 2000s, DNA array technology progressed rapidly as new methods of both production and fluorescent detection were adapted to the task. The knowledge of the DNA sequences of multiple genomes provided the raw information necessary to assure that arrays could be made which fully represented the genes in a genome, all the sequence in a genome, or a large fraction of the sequence variation in a genome. A gradual transition from spotting relatively long DNAs on arrays to producing arrays using 25 to 60 base pair oligos was noted during this time. The transition of oligoarrays was made possible by the increasing amounts of DNA sequence information. The use of oligos as opposed to longer sequences also provided an increase in specificity, for the intended binding target as oligos could be designed to target regions of genes or the genome that were most dissimilar from other genes or regions. Three basic types of arrays came into play during this time frame: spotted arrays on glass, in situ synthesized arrays, and self assembled arrays as shown in figure 2. Spotted arrays. In spotted arrays, polylysine coated glass microscope slides provided good binding of the DNA, and a robotic spotter was designed to spot multiple glass slide arrays from DNA stored in microtiter dishes. By using slotted pins similar to the fountain pens in design, a single dip of a pin in DNA solution could spot multiple slides, as shown in figure 1. Spotting onto glass allowed one to fluorescently label the sample. Fluorescent detection provided several advantages relative to the radioactive or chemiluminescent labels common to filter based arrays. First, fluorescent detection is quite sensitive and has a fairly large dynamic range. Second, fluorescent labeling is generally less expensive and less complicated than radioactive or chemiluminescent labeling. Third, fluorescent labeling allowed one to label 2 or more potential samples in different colors and co-hybridize the samples in the same array. As it is very difficult to reproducibly produce spotted arrays, comparisons of individually hybridized samples to ostensibly identical arrays would result in false differences due to array variation. However, a two-color approach in which the ratio of the signals on the same array is measured is much more reproducible. In-situ synthesized arrays. In-situ synthesized arrays involves chemical synthesis which combined photolabile protecting groups with photolithography to perform chemical synthesis on a solid substrate. In this production of arrays of 10 amino acid peptides and separately arrays of dinucleotides was demonstrated. Affymetrix demonstrated the ability to use this technology to generate DNA arrays consisting of 256 different octanucleotides. Affymetrix arrays are being used to detect mutations in the reverse transcriptase and protease genes of the highly polymorphic HIV-1 genome and to measure variation in the human mitochondrial genome by 1995 to 1996. Eventually, Affymetrix used this technology to develop a wide catalog of DNA arrays for use in expression analysis, genotyping and sequencing. A major advantage of the Affymetrix technology is that the DNA sequences are directly synthesized on the surface and only a small collection of reagents, that is the 4 modified nucleotides plus a small handful of reagents necessary for the deblocking and coupling steps are needed to construct an arbitrarily complex array. A method proposed by Blanchard and his co-workers in 1996 used inkjet printing technology and standard oligosynthesis chemistry to produce oligoarrays. In this method, inkjet printer heads were adapted to deliver to the 4 different nucleotide phosphoradamides to a glass slide that were pre-patterned to contain regions containing hydrophilic regions with exposed hydroxyl groups surrounded by hydrophobic regions as shown in figure 1. The hydroxylated regions provided a surface to which the phosphoramides could couple, while the surrounding hydrophobic regions contained the droplets emitted by the inkjets to define regions. This technology was eventually commercialized by Rosetta Informatics and licensed to Agilent Technologies, who produce these arrays at present. The inkjet array approach shares the advantage of Affymetrix and Nimblegen approach, in that one only needs to have a small number of reagents to produce an array. In addition, similar to the Nimblegen approach, the production of a new type of array only requires that a different set of sequence information is delivered to the printer. Hence, the inkjet array technology has been particularly useful for the design of custom arrays that are produced in low volume. Self-assembled arrays. An alternative approach to the construction of arrays was created by the group of David Walt at Tufts University and ultimately licensed to Illumina. Their method involved synthesizing DNA on small polystyrene beads and depositing those beads at the end of the fiber optic array, in which the ends of the fibers were etched to provide a well that is slightly larger than one bead. Different types of DNA would be synthesized on different beads, and applying a mixture of beads to the fiber optic cable would result in a randomly assembled array. In early versions of these arrays, the beads were optically encoded with different fluorophore combinations in order to allow one to determine which oligo was in which position on the array, referred to as the decoding of the array. Optical decoding by fluorescent labeling limited the total number of unique beads that could be distinguished. Hence, the later and present day methods for decoding the beads involve hybridizing and detecting a number of short fluorescently labeled oligos in a sequential series of steps. Applications of microarrays. Gene expression analysis. The predominant application of DNA microarrays has been to measure gene expression levels. In gene expression analysis, RNA is extracted from the cells of interest and either labeled directly, converted to a labeled cDNA, or T7 RNA promoter tailed cDNA, which is further converted to cRNA through the abovine amplification process as shown in figure 3. The methods for labeling of the cDNA or cRNA include incorporation of biotin labeled nucleotide, which is subsequently stained fluorescently labeled streptavidin, incorporation of fluorescently labeled nucleotides during the synthesis, and incorporation of a modified reactive nucleotide to which a fluorescent tag is added later, and a variety of signal amplification methods. The two most frequently used methods are the incorporation of fluorescently labeled nucleotides in the cRNA or cDNA synthesis step, or the incorporation of a biotin labeled nucleotide in the cRNA synthesis step. The labeled cRNA or cDNA are then hybridized to the microarray. The array is washed and the signal is detected by measuring fluorescence at each spot. In the case of biotin labeled samples, the array is stained post hybridization with fluorescently labeled streptavidin. Laser induced fluorescence is typically measured with a scanning confocal microscope. The intensity of the signals on each spot is taken as a measure of the expression level of the corresponding gene. Transcription factor binding analysis. Microarrays have also been used in combination with chromatin immunoprecipitation to determine the binding sites of transcription. In this, transcription factors TFs are cross-linked to DNA with formaldehyde and the DNA is fragmented. The TFs of interest are affinity purified using either an antibody to the TF or by tagging the TF with peptide that is amenable to affinity chromatography. Example, a flag, His, MIC or HA tag. After purification, the DNA is released from the TF, amplified, labeled and hybridized to the array. This technique is commonly referred to as chip for chromatin immunoprecipitation on a chip or microarray. The TFs often bind quite a distance away from the genes that they regulate. The design of the array and the size distribution of the fragment length are interrelated. In bacteria or yeast, the intergenic regions are fairly small and the small arrays used for gene expression work can be applied to chip. For mammalian genomes, the intergenic regions are large and the TF often bind many kilobase pairs away from the gene of interest. Hence, for mammalian genomes, oligoarrays with oligos spaced evenly across the entire genome are typically used for chip experiments. Genotyping. Microarrays have been widely used as single nucleotide polymorphism genotyping platforms. Several alternative approaches have been used to detect these SNPs, but the most commonly used are allele discrimination by hybridization shown in figure 4a as used by Affymetrix, allele specific extension and ligation to a barcode oligo which is hybridized to a universal array, the Illumina Golden Gate assay as shown in figure 4b, or approaches in which the arrayed DNA is extended across the SNP in a single nucleotide extension reaction, arrayed primer extension assay as shown in figure 4c, or the Infinium assay of Illumina as shown in 4d. Allelic discrimination by hybridization suffers background due to non-specific hybridization in complex genomes. In order to reduce this background, Affymetrix developed a PCR based approach to reduce the genomic complexity. SNPs are selected for their assay to be between restriction sites that are less than 1 kb apart. Genomic DNA is fragmented with a restriction enzyme, end repaired, and adapters of PCR are ligated to the fragments. PCR is performed under conditions that selectively amplify products of less than 1 kb in size. This method reduces genome complexity by approximately 50-fold and results in a corresponding increase in signal to the noise on array. Both the Affymetrix and the Illumina methods of SNP genotyping have been highly successful and are highly used. Today, SNP arrays capable of detecting more than 1 m different human SNPs are available from both vendors. Data standards and data exchange. With the exception of DNA sequencing, Microarrays were perhaps the earliest technology that allowed biologists to vast amounts of complex digital data. As the technology came into use, it rapidly became apparent that in order for others to be able to reproduce a given microarray experiment, a detailed description of the array, the sample, the protocols and the data analysis methods need to be available. Moreover, it also became apparent that access to the raw and processed data would allow others to perform analysis and meta-analysis on combinations of data that the original data producers had not conceived. To address these issues of reproducible science and data exchange, members of Microarray Gene Expression Data Society, now the Function Genomics Data Society, created the MIAME, Minimum Information About a Microarray Experiment Standards, for the description of microarray experiments and for the exchange of microarray data. These efforts influenced the creation public databases for microarray data and subsequent standards efforts in other areas. Limitations of DNA microarrays. At their core, microarrays are simply devices incredibly useful in a wide variety of applications, but they have a number of limitations. First, arrays provide an indirect measure of relative concentration. That is, the signal measured at a given position on a microarray is typically assumed to be proportional to the concentration of a presumed single species in solution that can hybridize to that location. At high concentrations, the array will become saturated, and at low concentrations, equilibrium favors no binding. Hence, the signal is linear only over a limited range of concentrations in solution. Second, especially for complex mammalian genomes, it is often difficult to design arrays in which multiple related DNA or RNA sequences will not bind to the same probe on the array. A sequence on that array that was designed to detect gene A may also detect genes B, C and D if those genes have significant sequence homology to the gene A. This can particularly be problematic for gene families and for genes with multiple splice variants. It should be noted that it is possible to design arrays specifically to detect splice variants either by making array probes to each exon in the genome or to exon junctions. However, it is difficult to design arrays that will uniquely detect every exon or gene in genomes with multiple related genes. Finally, a DNA array can only detect sequences that the array was designed to detect. That is, if the solution being hybridized to the array contains RNA or DNA species for which there is no complementary sequence on the RNA, those species will not be detected. For gene expression analysis, this typically means that genes that have not yet been annotated in a genome will not be represented on the array. In addition, non-coding RNAs that are not yet recognized as expressed are typically not represented on an array. Moreover, for highly variable genomes such as those from bacteria, arrays are typically designed using information from the genome of a reference strain. Such arrays may be missing a large fraction of the genes present in a given isolate of the same species. For example, in the bacterial species Agrigactibacter actinomycetocomitans, the gene content differs by as much as 20% between any two isolates. Hence, an array designed using gene annotation from a reference isolate will not contain many of the genes found in other isolates. The future of microarrays. Given the limitations of arrays mentioned above, it would be far preferable to have an unbiased method to directly measure the entire DNA or RNA species present in a particular sample. The advent of next generation sequencing technologies combined with the rapid decrease in the cost of sequencing has now made sequencing cost competitive with microarrays for all assays with the possible exception of genotyping. When the cost is similar, sequencing has many advantages relative to microarrays. Sequencing is a direct measurement of which nucleic acids are present in a solution. One need only count the number of a given type of sequences present to determine its abundance. Counting sequences is linear with concentration, and the signal to noise one can obtain by sequencing is only limited by the number of reads used for each sample. Sequencing is a relatively unbiased approach to measuring which nucleic acids are present in the solution. While sample preparation or different enzymes may bias sequencing counts, unlike DNA arrays, sequencing is not dependent on prior knowledge of which nucleic acids may be present. Sequencing is also able to independently detect closely related gene sequences, novel splice forms or RNA editing that may be missed due to cross hybridization on DNA microarrays. As a result of these advantages and the decreasing cost of sequencing, DNA arrays are being rapidly replaced by sequencing for nearly every assay that has been performed on microarrays. As the cost of sequencing is currently dropping by a factor or 2 every 5 months, it is likely that DNA arrays will be fully replaced by sequencing methods within the next 5 to 10 years. Let us now summarize the following discussion on microarrays. A microarray is a multiplex lab on a chip. It is a 2-dimensional array on a solid substrate, example a silicon thin film cell or glass slide, that assays large amounts of biological material using high throughput screening, miniaturized, multiplexed and parallel processing and detection methods. DNA microarrays are a group of technologies in which specific DNA sequences are either deposited or synthesized in a 2-dimensional or sometimes 3-dimensional array on a surface in such a way that the DNA is covalently or non-covalently attached to the surface. Grunstein and Hogness created the DNA array with the colony hybridization method. In the late 90s and 2000s, DNA array technology progressed rapidly as both new methods of production and fluorescent detection were adapted to the task. 3 basic types of arrays came into play during this time frame: spotted arrays on glass, in-situ synthesized arrays and self-assemble arrays. The predominant application of DNA microarrays has been to measure gene expression levels. Microarrays have also been used in combination with chromatin immunoprecipitation to determine the binding sites of transcription and single nucleotide polymorphism genotyping platforms. Before we attend the next unit, please try to answer the following questions. Write an overview on the early history of microarrays. What defines an microarray? Briefly explain different types of microarrays. Define Affymetrix technology. Discuss different types of microarrays. References: Introduction to Bioinformatics by Atwood and Parry Smith, Pearson Education. Bioinformatics, a beginner's guide by Jean-Michel Clavier, John Wiley and Sons. Structural Bioinformatics by Philip Bone and Helge Wesing, Wiley. Bioinformatics, Methods and Applications by Rastogi SC, Mendirata N and Rastogi P. Prentice Hall of India Private Limited, New Delhi. Hope this session was enriching. Thank you for watching this session and goodbye.